Hello, my lovely anatomists and physiologists. Welcome back to our videos on cardiovascular system. We are ready to enter into our chapter 20, and we're going to be talking in chapter 20 about blood vessels, movement of blood through those vessels, how we get oxygen delivered to our tissues, and then regulation of the whole system. So we're going to be putting a lot of pieces together here. To start out in this conversation about blood vessels, let's remind ourselves of a few things that we've already said. So when we talk about blood vessels, we have two main categories, right? We have the artery and we have the veins. And when we talk about the arteries, remember, I am encouraging you to use the letter A to remember arteries are always away from the heart. And then the veins are toward the heart. And so in this way, you'll always be able to keep up with whether you're looking at an artery or a vein if you're able to trace the blood as it moves through. Now we're going to start in this video looking at what we can describe as shared features. So these are things that arteries and veins are going to have in common. Okay. And so the first thing that we want to pay attention to is that blood vessels have blood vessels. Oh my goodness, this is so amazing. Think about it a minute. Blood vessels, especially big ones, have their own blood vessels. Whoa, mind-blowing, right? Okay, so we talk about the blood vessels of blood vessels as the vasa vasorum. So these are going to be the blood vessels that are going to serve larger arteries and veins. And so as you investigate your lab work, you're going to be looking at examples of several arteries and veins. And so you'll get an idea about what are some of those bigger ones. But for us already in this lecture, we've talked about, for instance, the aorta and the vena cava. So clearly these are big ones that would have their own blood vessels. And then we also see that they're going to be innervated. And so we have what's described as a nervy vasorum. And this is talking about super tiny sympathetic nerves that's going to control what we call contraction or constriction, vasoconstriction, and dilation or vasodilation of the smooth muscle that we find in our veins and our arteries. I should have just said in our vessels. And the other thing that we're going to see in common for all the blood vessels are the three tunics. So three tunics, really, this is like the three layers. Tunic is named as in like clothing, right? So like an outside coat. Oh, let's do that. Okay, so the tunica externa would be like your coat. You know, like your winter coat. And then the tunica media would be like your sweater. And then the tunica intima would be like your undershirt. Okay, so what we're saying here is the tunica externa is the outermost layer of the blood vessel. The tunica media is the middle, and the tunica intima is the innermost. And we're going to look at characteristics of each of these in terms of the kind of general information. And then we will have a whole video dedicated to just the differences between arteries and veins. When we talk about this outermost coat, tunica externa, this is going to be made of connective tissue. And this fits a pattern you should have been seeing throughout the course. Anytime we study little organs, structures, we see the outermost layer is a connective tissue. So it's at once like connecting it back to the body and then also making it separate in some way. Tunica media, this is the smooth muscle layer. So when we do talk about constriction and dilation of vessels, we're talking about manipulating or changing the activity of this layer here. And then your tunica intima, which is like our undershirt, right? So this is going to have connective tissue, but also epithelial tissue. And this should make sense based on like your understanding of tissues in the body. You know, I like to always describe the epithelial tissue as lining the innermost pieces and parts of our body. And so that would make sense here. Going back to the tunica externa, our outer coat made of connective tissue, we're going to see that it's mostly going to be collagen and some elastic. And this is going to vary based on vessel type. I'll say some elastic fiber. Remember that your collagen fibers are your super strong. They're going to be able to bend without breaking. And your elastic fiber is stretchy, so it can expand and recoil. So think about that rubber band. And what we'll see is that the tunica externa will actually, in this case, blend with the surrounding connective tissue, helping to really like anchor the blood vessel in place. So I'm going to say anchor vessel. Our textbook uses the terminology of like holding the vessel in position, but essentially we're decreasing movement of that vessel. So think about it. Your body is very active. Your body is so active. So our skeletal muscles moving, we have all kinds of muscle inside our organs that's moving, the vessel itself has muscle that's moving. If we allow a lot of movement of the vessel as a whole, that's going to disrupt that blood flow. That's going to disrupt that blood flow. So really anchoring it in with the connective tissue is going to help stabilize that blood flow to some extent. Taking a look at the tunica media, remember this is our smooth muscle layer. We're going to see that we have a circular layer of smooth muscle, and this is the layer that will actually change diameter, right? So when we have circular muscle, when we contract that muscle, we're constricting or making that passageway smaller. And when we relax that muscle, we're dilating that vessel. But we also have longitudinal muscle, which we talked about that in terms of our blood clotting mechanism we talked about during the vascular spasm. The circular muscle was helping to kind of close off that opening. The longitudinal muscle was sort of like pulling it back into the tissues. So we talked about that just a little bit there. We're going to see when we talk about the changing of diameter, we have what's called vasoconstriction. When we talk about the vasoconstriction, that's going to reduce the artery, which is going to then reduce blood flow. We also have what's called vasodilation. That's the opposite. So that's going to increase the artery diameter and increase the blood flow. And then we have what's called venoconstriction. And this is specifically talking about changing the diameter of our veins. And so here, this is reducing the vein diameter. So just know when we talk about vasoconstriction and vasodilation, we're really talking about the artery. We're really talking about the artery. We'll see too that we're going to get local controls involved in all of this movement here. So we're going to have local control. We're going to see endocrine control. And we've really talked about it's the sympathetic nervous system primarily that's going to be involved in controlling the smooth muscle of the vessels. And then last we have our tunica intima. Tunica intima. It's like our undershirt. So this is the innermost lining of our blood vessel. And so here we're going to see, let's start with the basement membrane. This is going to be the connective tissue layer. So the basement membrane is going to be connective tissue. And it's going to be strong. It's going to be flexible. And it's going to be permeable. So when we do talk about the need for material to cross in and out of the bloodstream, we'll see that it can move across that basement membrane. We also are going to see what's described as the endothelial cell. So we sort of mentioned these before when we were talking about, again, blood clotting and when we talked about the vascular phase. The endothelial cell is the simple squamous epithelial tissue that's lining the entire cardiovascular system. So it's the innermost lining of all of your blood vessels, as well as the chambers of your heart. Endothelial cells are the simple squamous epithelial cells that line the entire cardiovascular system. So that's going to be true for both all the blood vessels as well as the chambers of the heart. Now, what we will see is that they can actually release two things. So they can release what we've already talked about as endothelium. Make that a little bit easier to read. Endothelium. And if I can get my highlighter, let's highlight that. Endothelium is a chemical released by endothelial cells, so that name makes sense. And this will be an example of a local factor. It's going to constrict blood vessels. So we saw that already again in that vascular spasm, that vascular phase, and we saw it continuing. Yeah, let me just stop there. And then we'll also see that endothelial cells can release nitric oxide, which we'll abbreviate NO. And nitric oxide is going to be a vasodilator, which we know then means make that blood vessel larger. So these are going to be examples of some local controls that would be altering the diameter of that blood vessel. Up next, let's look at the differences between arteries and veins specifically. So stay tuned. As always, take care of yourselves and each other.